Hello, in this video we will discuss about the glycolysis and gluconeogenesis and their significance we will understand. So remember, the glycolysis and gluconeogenesis formation without using oxygen is known as anaerobic respiration. So the fat state during when we will eat carbohydrate will go into the stomach, and digestion of the carbohydrate into the simple sugar will absorb is known as absorption after the digestion. And this hyperglycemia will occur in the blood circulatory system, will lead to stimulate the pancreas. The beta cell will stimulate to produce insulin, and this insulin will bind with a specific tissue, the containing cell. And after this containing the cell, after the absorption, this receptor will bind with the insulin receptor, lead to function for glycolysis if the oxygen is not present. If oxygen is present, so the glycolysis will lead to produce pyruvate to produce the ATP from the electron transport chain in the mitochondria, while the glycogenesis will occur in the muscle and the liver. So remember, this is the blood vessel. So this is the assimilation part in the cell is known as assimilation. While during the fasting state, the here is the stomach with duodenum and pancreas, the circulatory system. So hypoglycemia will occur when we will not eat, will stimulate the alpha cell of a pancreas, will produce the glucagon, which that will lead to decrease the glucose, and the epinephrine and the glucagon will bind, for example, with the liver and liver and adrenal gland, kidney, which that produce the gluconeogenesis formation will produce the glucose, and hypoglycemia no insulin will produce, and in this way the glucagon will move into the blood circulatory system. So remember, this is the glucose will enter into the cell for the glycogenolysis occur, for example, in the other cell, and glycogenesis will occur, and during the no assimilation from the digestion. So let's begin to understand the glycolysis. So here is the sucrose contain fructose and glucose, which that invertase enzyme and water use to split two molecules, the glucose and fructose. So remember that things. And after this separation, we are understanding the glycolysis. So remember. So this invertase and water is using to produce the two molecule glucose and fructose. This glucose will lead to phosphorylase to add the phosphate group, will go either into the glycogenesis or go further into the glycolysis process, while the phosphorylase will use the ATP, and the fructose and the glucose will use ATP and phosphorylase enzyme to add the phosphate group in the carbon number 6. So when the carbon number 6 basically containing the phosphate group is known as the glucose 6 phosphate or fructose 6 phosphate formation. So remember, in this way the glycolysis will run. But remember, the starch when we will eat will digest and convert into the simple sugar, and the simple sugar undergo the phosphorylation due to the phosphate group phosphorylase enzyme to produce the glucose 1 phosphate. So remember, this reaction is basically reversible. It can be converted due to the mutase enzyme, the fructose 6 phosphate mutase enzyme or glucose 6 phosphate mutase enzyme, to convert either go into the glucose or fructose. So in this way, here is the starch which that will be digest, and the starch basically the amylase enzyme is used during the digestion from the pancreas, release exocrinally. But in this case, when go into the cell, it will convert by the phosphorylase enzyme to the phosphate 1 phosphate glucose 1 phosphate, and the phosphoglucomutase enzyme used to produce the glucose 6 phosphate. In this way, the phosphate group will convert from the 1 to 6 carbon number. So anyhow, after the phosphofructokinase enzyme use with the ATP to convert the fructose 1 and 6 bisphosphate, this fructose 1 and 6 bisphosphate will be split into two molecules with containing 3 carbon, and it is the under the action of aldolase enzyme to produce the aldo sugar. So the triose phosphate isomerase enzyme used to interconversion into the glyceraldehyde 3-phosphate or glyceraldehyde 3-phosphate or phosphoenol pyruvate. So anyhow, in this way here you can see the carbon number three containing the phosphate group. So that is why it is known as the glyceraldehyde 3-phosphate. But the 3 glycerate phosphoglycerate, basically here you can understand. So anyhow, here is the glyceraldehyde 3-phosphate dehydrogenase enzyme used when add the phosphate group and the NADH used due to the dehydrogenase enzyme to release the NAD plus to produce the 1,3-bisphosphoglycerate. This 1,3-bisphosphoglycerate is the character, the carbon number 1 and 3 containing phosphate group. That is why it is known as the 1,3-bisphosphoglycerate. After this 1,3-bisphosphoglycerate will under the action of the phosphoglycerate kinase enzyme used with the ADP due to the using of the ATP will basically to produce ATP, and in this reaction ATP will produce 3-phosphate glycerate because the carbon number 3 containing phosphate group. After this phosphoglycerate mutase enzyme used to mutation in this formula in this molecule to shift the phosphate group into the carbon number 2, and in this way it is known as the basically the phosphoglycerate 2-phosphate. And enolase enzyme to release water is dehydration, and the dehydration reaction will lead to produce the another compound. The basically it is carbon number 2 contain phosphate group, but it's different. So in this way the pyruvate kinase enzyme used to, release the ATP, and in this reaction the ATP will produce under the action of pyruvate kinase enzyme to produce pyruvate. This pyruvate is 2 because the splitting of glucose into 2 molecule 3,3. So in this way we can say that is pyruvate is a 2 molecule containing 3 carbon. So after this, if pyruvate will not go into the mitochondria and requirement of the glucose in the blood circulatory system will more, so in this way the gluconeogenesis process will be occur. This reaction is due to the fasted state because the glucose is not intaking. So that is why the gluconeogenesis formation will lead to produce the glucose into the blood circulatory system. That is why important things you should need to understand for isoglycemic level from the hypoglycemia. So the biotin dependent pyruvate carboxylase enzyme used for the pyruvate conversion into the oxaloacetate. This oxaloacetate use the GTP to convert the GDP and carbon dioxide release, and to in the presence of phosphoenol pyruvate to produce the phosphoenol pyruvate. This phosphoenol pyruvate is basically 3 carbon containing compound which that contain a phosphate group. So that is why it is known as the phosphoenol pyruvate. And this formula in the given in the diagram I will show here. So in this way this phosphoenol pyruvate, which that is basically you can understand it is the reverse reaction is going on. And in this way when water will be added in the presence of enolase enzyme rather than the aldolase. So enolase enzyme when add, so the 2-phosphoglycerate formation will be occur. 2-phosphoglycerate mean the carbon number 2 containing phosphate group. So that is why the 3 carbon containing compound which that is 2-phosphoglycerate, because here you can understand. And after this, the 2-phosphoglycerate, in the presence of phosphoglycerate mutase enzyme will lead to mutation to cause 3-phosphoglycerate because the carbon number 3 containing phosphate group and the phosphoglycerate kinase enzyme used for the 3-phosphoglycerate to convert phosphoglycerate kinase to convert the 1,3-bisphosphoglycerate. So the phosphoglycerate kinase here is enzyme is using to convert and the addition of the further phosphate group into the carbon number 1. It means that 1,3-bisphospho 2 phosphate group, the bisphosphoglycerate formation will be occur. And in this way the carbon number 1 and carbon number 2 carbon number 3 containing phosphate group with 3 carbon containing molecule is known as a 1,3-bisphosphoglycerate because the glycerate containing 2 molecule of phosphate to a phosphate group. So in this way, the triose phosphate isomerase enzyme used to produce the dihydroxyacetone phosphate DHAP. This dihydroxyacetone is basically similar after the splitting in the presence of aldolase enzyme to produce that enzyme and that compound similar, but the reaction is same, and the reaction is same going to reverse. So the aldolase enzyme again this aldolase enzyme used to produce again that fructose which that is that produced during the glycolysis. So it reverse, it will move upward, so the aldolase to reverse and back to produce the 1,6 bis phosphofructose. So in this way, the again phosphate group will be released in the presence of water. So the hydration reaction will lead to fructose bisphosphate phosphatase enzyme used to produce the fructose 6 phosphate. So in this way, the glucose 6 phosphatase isomerase enzyme is used to produce the glucose 6 phosphate. This glucose 6 phosphate is basically the phosphoglucomutase enzyme used to produce the glucose 1 phosphate. Glycogen precursor is that it either go into for glycogenesis or the glucose formation into the blood circulatory system for normal function. But remember the glucose 1 phosphate which that is required for the glycogen synthesis. So in this way the glucose 1 phosphate in the presence of the phosphoglucomutase enzyme which that will produce the glucose 1 phosphate, the glycogen precursor, while on the other hand the glucose 6 phosphatase isomerase enzyme used to produce this glucose 6 phosphate and under the action of water to release the phosphate group to produce the a simple sugar, the glucose. This glucose will go either into the cell or into the blood circulatory system for normal blood glucose, the isoglycemia, rather than the hyper or hypoglycemia. So in this way, the glycogenesis can be possible as well as the glucose formation can be possible through gluconeogenesis during the exercise. During the exercise, the gluconeogenesis can be possible, while the glycolysis due to the anaerobic respiration that is produce pyruvate to use for the mitochondria to produce more ATP during the Krebs cycle and electron transport chain. But electron transport chain and Krebs cycle we are not discussing in this video. So you can imagine that the glycolysis is the breakdown, the catabolism, while the gluconeogenesis is the anabolism. During the fasted state, the anabolism will occur for the normal glucose level. So thanks for watching. Bye.